Hello, everyone. I'm Christine Lambert, Senior Scientist here at PacBio, with today's topic focusing on scalable workflow for constructing HiFi libraries. So today I'll be sharing with you a workflow that was created in collaboration with Children's Mercy Kansas City and Rabu University to sequence and analyze over 100 patient samples representing 80 singletons and 7 trios that were negative with the short read whole genome sequencing. And our goal with the collaboration was to create a workflow, HiFi library workflow that is scalable and easy to use. So the new scalable HiFi library construction workflow is now available on our PacBio website. This is the procedure and checklist for preparing HiFi SMRTbell libraries using SMRTbell Express Template Prep Kit 2.0. So you could click here and it will provide you a published protocol. So before you start your experiments, it's important to note that HiFi sequencing requires high molecular weight DNA. And so before you start, we recommend reviewing your technical note for how to prepare DNA for PacBio HiFi sequencing. In this technical note, it provides recommendations for DNA extraction and quality control. So this is a link to our tech note. So in efforts to make more workflows more scalable, we've developed a workflow on these 3 different aspects of the workflow: DNA shearing, library construction, and size selection. And the goal of optimizing these workflow was to enable preparation of samples in batches up to 16 by manual or automated methods, to enable consistent library yields across samples, and to enable size selection of multiple samples per run. So the steps in the workflow that we optimized were, of course, the DNA shearing on the Megaruptor 3 system. Using this newer system allows shearing up to 8 samples per run, and the processing time takes about 75 minutes. And more importantly, the shearing distribution across the 8 syringe hydropores are quite consistent. And by having consistent shearing will give you consistent yield, will give you consistent sequencing performance across different samples. So this is very critical. The second part is the DNA construction. So we've updated the protocol so you could process more than 8 samples at a time, and by using strip tubes and multi-channel pipettes. And the protocol is now amendable to automation, which I will go over later in this presentation. For size selection, we've implemented the Pippin HD from Sage Science for size selection, which will enable you to process more samples per run. So you could process up to 20 samples, 10 samples per cassette, and the runtime for size selection is greatly reduced down to 2 hours. Addition to streamlining the protocol, we've also reduced the input amount of genomic DNA required. So we've reduced it to as low as 5 micrograms versus 15. And this 5 micrograms of genomic DNA is sufficient for greater than 4 SMRT cells. So advantage of the Megaruptor 3 system is the ability to shear the genomic DNA to a narrow size distribution. And so in our procedure, we've developed a method to just shear genomic DNA to a quite tight size distribution across different samples. Our recommendations for Megaruptor 3 would be to start with 5 micrograms of genomic DNA and a 60 microliter elution volume of a final concentration of 83.3 nanograms per microliter. And it's important before you go forward with your library preparation that your samples must be in this QIAGEN elution buffer. So if not, then we recommend a buffer exchange using the Ampere PB beads purification. In order to generate a tight size distribution, we recommend using a 2-cycle shearing process in the same hydropore. So first you would take your DNA and shear it on the Megaruptor at speed 31, followed by a second shear at speed 32, all done in the same hydropore. The runtime is about 75 minutes. And as always, optimizations may be necessary to target your desired distribution for your project. So this compares 2-cycle versus single-cycle shear, and we can see with the 2-cycle shear, the size distribution is much tighter. So on the left here is the femtopulse profile comparing 2 versus 1-cycle shearing, and you can see that the 2-cycle shearing is much tighter. There's a tighter distribution, and the amount of fragments less than 30 kb is much reduced compared to a single-cycle shearing method. And so this, in this case, in this example, this is the same DNA just sheared with the two different methods. On the right here is the femtopulse profile overlay of 15 human samples that were sheared using the 2-cycle method. And you can see that with the different human samples, the size distribution is quite consistent across the 15 different human samples tested with this 2-cycle shearing method. Let's see. And so for so this is for the previous slides where the 2 libraries were sequenced. This is the HiFi Relength comparison, comparing the 2-cycle versus the single-cycle shearing. You can see with the 2-cycle shearing improves the medium HiFi Relength quality. So you could see here with the medium HiFi Relength quality Q31 for 2 cycles versus a single cycle of shearing Q21. Then you can see that the distribution is quite different. With the 2-cycle shearing, there's a very little fragments, less than 30 kb, compared to a single cycle shearing where you could see there's about 8% of the reads are 30 kb. And also other advantages of using this 2-cycle shearing method is the HiFi yield is higher, and there's more consistent loading across samples, and the required on-plate concentration is lower. So with our new Streamline protocol, since 16 samples can be completed in 2 days from DNA shearing to size selection, you could use PCR tubes using multi-channel pipettes, and to further streamline the protocol, you could prepare master mixes for the reactions. And below here is the hands-on time and the walk-away time. And so for the hands-on time, it takes about 30 minutes or 60 minutes for processing 16 samples for the library preparation. So the bottleneck in the workflow is the size selection. So we're now recommending size selection with the PipInHT. The PipInHT is higher throughput, so you can process up to 20 samples in 2 hours. The other systems, Sage systems, Sage ELF and Blue Pippin, do a really good job at removing fragments less than kb, but the PipInHT is higher throughput. And what we recommend for the cassette definition would be a 6 to 10 kb high pass protocol using marker 75E to remove these shorter fragments. The percent recovery is higher, which allows reduction of input genomic DNA into your projects. So here below is the femtopulse profile comparing a pre and post size selection. And you can see with these human non-size selected libraries, there's quite a lot of fragments less than 10 kb. And when you size selected using the PipInHT system, the libraries are quite tight and narrow, which is what you want for HiFi sequencing. And the fragments are completely removed, the smaller fragments, which is great. So with the new Streamline protocol, we've worked with Perk and Elmer to transfer the protocol to the Sciclone G3 system. And so by doing this, we can automate up to 96 samples. The input would be the sheared DNA, and then the system you can perform the automated enzymatic V2, Express V2 library prep reactions and Ampere PB purifications. And the output would be SMRTbell libraries ready for size selection on the PipInHT system. So this is the instrument, the Sciclone G3 system by Perk and Elmer. So this is an example of a HiFi library sequence on the Sequel IIe system. The typical HiFi yields are between 25 to 35 gigabases per smart cell. And the image here on the left is a HiFi library that's sized by the femtopulse, which is approximately 17 kb. The image here shows pre and post size selection by PipInHT, and you can see that the PipInHT does a really good job of removing fragments less than 10 kb. It generates a really narrow size distribution, which is good for HiFi sequencing. The distribution of this library is consistent with the distribution of the HiFi reads shown here in this insert length density plot. The CCS reads, which are shown in red, are between 10 to 20 KB, and which are also consistent to the HiFi Relength distribution here, which is a 17 KB. In this example, this library was loaded at 40 picomolar on plate with a P1 of 70%. With 30-hour movies, the HiFi yield is around 34 gigabases with a median HiFi quality of Q30. So in conclusions, we recommend shearing the DNA with the Megaruptor 3 system with the 2-cycle shearing, which will increase size consistency. On plate, for size selection, we recommend using the pipet HT, and you can manually prep up to 16 samples in 2 days or 32 libraries in a week versus the old prep where you could only do 3 libraries approximately. And we've automated our workflow so you could process up to 96 samples on the Sciclone system. And in addition, we've reduced the input DNA from as low as 5 micrograms of starting material, which will generate 4 or more smart cells. So I invite you to explore other on-demand presentations. Ginny Ekholm will be presenting the introduction to HiFi sequencing for rare disease research, and then William Rowell will be presenting the data analysis for this workflow. And I encourage you to learn more about HiFi sequencing for rare disease research by exploring our website. And if you have any additional questions, please contact us at pacb.com/scientists. Thank you for your time.